guys, welcome to Biomix Hub. In this today's video, we are going to discuss about one repair mechanism of DNA, that is a mismatch repair mechanism. So during the DNA replication, there is synthesis of the DNA by seeing the parent strand. A daughter strand is being synthesized by use of a enzyme which is DNA polymerase. So during the synthesis, there is certain mismatch. The DNA polymerase incorporates some incorrect nucleotide. So this nucleotide needs to be recognized and it needs to be removed by the synthesis of the correct nucleotide. So this is being done by this repair mechanism, mismatch repair mechanism. Okay, so during this mismatch repair mechanism, there are certain proteins, so we can tell that certain enzymes which are present. So the first enzyme is MUT-S. MUT-S. This work of this MUT-S is it recognizes the incorrect nucleotide. What happens in case of a DNA? Adenine base pairs with thymine and guanine base pairs with cytosine by the help of 3 hydrogen bond. So we can see here this is the parent strand. This is the parent strand in which there is thymine. This thymine needs the DNA polymerase during the synthesis. It needs to incorporate adenine, but by mistake there is incorporation of guanine here, due to which there is distortion. The work of MUT-S is it will recognizes this place where the mismatch is taking place. We can see that the MUT-S which is present, it is in the form of dimer. It is like a clip. What it does, it scans the DNA. It scans the DNA and it recognizes a particular place where there is a mismatch, and this is a ATP dependent process. Here ATP is being used. After that, it recruits some other proteins which is known as MUT-H and MUT-L, and this is also a ATP dependent process. The work of MUT-H is it creates a nick. Okay, it creates a nick in this region. This MUT-L which is present, it comes and binds in the parent strand, and the work of MUT-H, it comes and bind near the daughter strand. It will do its work in the daughter strand. It will create the nick in the daughter strand. After the synthesis, after the cut, there is requirement of another protein which is known as UVRD. This UVRD, it is a helicase. It opens the DNA. It opens the DNA so that the exonuclease can come and it will do its work. It will cut the DNA. So after the cutting, there needs to be, this gap needs to be filled. So this filling of the gap is done by DNA polymerase. So work of DNA polymerase is for the synthesis of the DNA, and for that it requires 3' OH group. Here there is a 3' OH group where the DNA polymerase will come and bind here, and there will be synthesis of this DNA. Okay, at the end there will be involvement of DNA ligase for sealing the gap. In this way, the incorrect nucleotide is being replaced by a correct nucleotide. Now let us see with this help of this other diagram how this MUT-H works. So the MUT-H which is present, it is present near the GATC sequence. Okay, this adenine which is present, it is in the methylated form. It is in the methylated form in case of parent strand, but in case of daughter strand, it is not methylated. In this way, this MUT-H, it recognizes the daughter strand and it creates a cut in the daughter strand. So the MUT-H which is present, it is present near the GATC sequence and it is in the inactive form. It is in the inactive form. Like we will see a DNA. Here there is a GATC sequence. Okay, so here there is MUT-H, and this MUT-H now it is in the inactive form. If there is recognition of some of the distortion by MUT-S, then there is a bending by which the MUT-H and MUT-S, that they will be in contact. So for creating the contact, that first thing there is a bending, and second thing there is involvement of MUT-L. Okay. So after the bending, it creates a connection, or there is a connection between them. After that, this MUT-H which is present, it will be activated and it will cleave the DNA. Okay. After the cleaving, there will be certain exonuclease which will come and bind. Here we can see there is a exonuclease which comes and bind, and here there will be degradation of this DNA, and further the DNA will be synthesized by DNA polymerase. Okay, and there will be synthesis of correct nucleotide. We can see here guanine is base pairing with cytosine. Here in this diagram there was incorporation of some other nucleotide, but in the first diagram we saw that there is in place of adenine there was cytosine. Here there was some other mismatch, like C is being this C is being present here. In this place thymine was present. Okay, so here we can see that how this is being recognized, how this how there is identification whether it is a daughter strand or a parent strand. In case of a parent strand, here there is a methylation in the adenine, but in case of daughter strand, here there is no methylation of adenine. So the mute H recognizes this place and it creates a cut. So there are involvement of some exonucleases. So there can be two types of exonuclease, which were they are the 5' to 3' exonuclease or 3' to 5' exonuclease. We can see here that the mute S is present here. So this is the place where the distortion or the nucleotide is showing the mismatch. After that, the mute H and mute L is being recruited, and if it cuts in this region, that is 5' here, that is 5' to 3' exonuclease, so it will cut in this region. Okay, so after the cutting, here there will be degradation. It is from 5' to 3'. That means it will this exonuclease which is present, it will be carried in this way. Okay, so in this way the DNA, it will be removed. After that, the DNA polymerase will come and bind in this region. What is the other name of 5' to 3' exonuclease? It is exo 7 or rec J. Okay, rec J. This is the other name of 5' to 3' exonuclease. Okay, so what if there is some other nucleus like 3' to 5' exonuclease? So here there is a mutation, or after this there is a mutation in this region, there is a mutation, or we can tell that there is mismatch in this region, but the involvement or the recruitment of the mute H is taking in this place. Okay, so during this time it needs to be removed. If this work in this way, there is no distortion in this place, so it needs to go backward. So here the 3' to 5' exonuclease work, so it can degrade this place. Okay, it can degrade this place. So now here there is involvement of DNA polymerase and it fills a gap. So what happens if the mutation, wherever the place of mutation is taking place, the mutation place will be constant, but where the mute H is present, in which GATC the mute H is present, it will be differ. So accordingly there will be involvement of either 3' to 5' exonuclease, or there will be involvement of 5' to 3' exonuclease. It will be decided. In this way the DNA repair mechanism work. So in case of mismatch repair mechanism, what are the enzymes involved? The first enzyme was mute S, which was a dimer, and it was recognizing the DNA by scanning the DNA. The second was the mute L and mute H. The mute H, it is a CSIR. It is working as a CSIR. It cuts the DNA, and it is inactive when it is not in contact with mute S. When it is in contact with the mute S, with the help of bending, it will be activated and it cuts the DNA in the daughter strand. Then there is involvement of UVRD, which is a helicase. It is a helicase. Then DNA polymerase 3 comes and it synthesizes DNA, which is being further sealed by enzyme ligase. Okay, in this way the DNA repair mechanism works. How the mismatch repair mechanism takes place. In the next video we will discuss about the other repair mechanism. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.